And now we move on to our graduate talks from more IDO alumni. Well, it's always good to see more and more IDO every year coming back and giving back to our presentation space. So we'll get our first one underway. Please welcome Jay Wong.
So good morning, everyone. Thank you for coming, and I'm grateful to be up on the stage talking about the internship that I conducted with Swiss TPH, just imparting some lessons learned from the implementation of this app. And so I was going in and technically assisting the Ivorian healthcare professionals in using the app. So let's begin. So, rabere means rage in Greek, and rabus means to do violence in Sanskrit, Indian. And there's no wonder why this is the rabies virus etiology, because it is a really savage disease that has been feared by mankind for thousands of years. So this dog is probably rabid and was filmed in Morocco. And the virus has now entered the brain into its salivary glands and ready to infect the next victim. So not only does it make dogs furious, it is also a very stealthy virus, because as soon as symptoms are apparent, death is 100% inevitable. But it is entirely treatable if treatment is given as soon as possible and compliance is full. However, lots of people in the world have inadequate access to healthcare and resources, which is why 59,000 deaths per year occur, mostly in Africa and Asia. If 70% of dogs are vaccinated, theoretically herd immunity could be reached, and the viral transmission could be interrupted at its source, which is the dog reservoir in most cases. And a One Health approach is really pivotal to combating this disease. And across the world, the veterinary sector and the public health sector do work together to combat this disease. A global 0 by 30 strategy was adopted by the Tripartite Alliance, including WHO, FAO, and WOA, to end dog-mediated rabies by 2030. So in 2016, Swiss TPH, and in partnership with the Swiss Scientific Research Centre in Côte d'Ivoire, implemented this project to estimate the rabies burden. And a few limitations were found. This includes vaccine wastage, lack of intersectorial communication, and follow-up constraints. And so in 2020, there was a block rabies project that was launched to really improve this. And the main aim is to really ensure that patients are able to receive their treatment as fast as possible. This is achieved by improving the communication, digitizing the health records, and transparently managing vaccine stocks, all on a secure blockchain platform. And I was tasked to really come in and ask, why are people not adopting the app? Like, what are the barriers, and what are some potential solutions to combat this? So I was based in Abidjan, and I was working with the local project management team and traveling between San Pedro and Boaké, the two pilot sites, where I'd work with the vets at Mira and the public health nurses in INHP. And so what I facilitated were workshops in April and May, and this included coaching sessions, one-on-one sessions, where we would really help the actors to use the app along their journey, because competencies were differing, ages were different, for example. And in between, we also really encouraged that they use the app on the tablets using fictive cases before the real productive system was put in place. So some type of data collection methods that we were adopting were questionnaires, both open and closed questions. We also asked actors' perceptions along the evolution of the intervention, application update requests, time to complete each application section, and some login history. And so what I was doing then, over the 4 months I was in Côte d'Ivoire, I was helping to quality assure and test the application, really co-designing with the end users to best tailor it not only in French, but to the Ivorian working context. And then feedbacking to the management team and also the programmer to best tailor it to their needs. So, introducing the apps a little bit. This is the main app is the Block Rabies app, which was digitizing the vaccine supply chain, the patient health records, the vet health records, but also the dog diagnostic results all on the same platform. And there was a vaccine scanning app as well, using a QR code where the vaccine stockers could do a whole inventory check, and then it would be uploaded onto the app, where the nurses could then select the vaccines that were being used on the patients, and we could kind of trace the vaccine stocks. So in all, there were about 150 update requests that were collected. So if we take, for instance, a person comes in with their dog that has bitten someone, what happens as soon as they come in is that the dog is given a rapid diagnostic test and put under surveillance for at least 10 days. And it's only once they start developing symptoms that the dog's head is cut off and sent to the laboratory for an immunofluorescence test. So really, within the application, the surveillance section needs to come before the diagnostic reports. So this was one of the updates. And on the index page, for instance, in order to get to the case file quickly, for instance, when someone comes for their second vaccination, it's great that more filtering options are available. So, for instance, with animal name or property owner, these were kind of the things that were requested. And also, the programmer was from Basel, so German-speaking and didn't speak French. So sometimes things got lost in translation, and we were also doing some translation changes. And I'd use a PO editor for that. Anyway, so this was a great result that we saw, was that with the scanning app, it took the vaccine stockers less than 5 minutes to scan 300 rabies vaccines. And this is a whole inventory check that usually takes a whole day. So they were really happy with this and were saying that they would want to adopt this right away. So also along the course of the workshops, through your questionnaires, we were asking, like, what advantages do you see within this application? And the major one was that data is securely stored. And for the nurses, they believed that better patient management was a great bonus, and also, and then for the vets, it was that data is shared instantly between the institutions. So we also saw that confidence increased for end users over time. For instance, the nurses, 47% more nurses felt they had good proficiency of the application in May. So in order to assess whether workshops were being the need for more workshops and whether they were going well, we timed how long it took each user to go through their workflow, and we saw that over time the time it took decreased, which is great, except for a couple of outliers, and this might be due to the fact that they were getting distracted, and so accuracy of the timing wasn't optimal. And I was, working with quite a few people at the same time, so potentially that was also a reason for that. We also saw the same trend for the veterinarians, and we saw that, the time also decreased to complete the application sections. So we identified barriers that were technical and organizational, and the main issue we saw actually was that internet connection issue was a problem. 58% mentioned it as their biggest worry. And so during the workshops as well, there were some power cuts, which meant that we couldn't use the application during the workshops. So really, a generator would be a good solution, or creating an offline function where drafts could be saved, and drafts could be saved, and then when online again, it can be saved. It can be put on the system, and the interface could also be a bit more intuitive. So we were making sure that the least amount of clicks occurred, and then account privacy was also something seen. We'd like to potentially give a two-factor authentication. And so more equipment was also requested. We'd like to distribute it more evenly through the institutions. Lack of trainers was mentioned by a couple, and longer workshops and promoting really peer learning would be a solution there. And making sure that incentives are carried on and improving the communication. So in conclusion, with training and practice, the actors improved their use of the applications. Peer learning should be encouraged, as well as offering continued coaching sessions. The majority believed actually that eventual transition could be achieved by the end of the year after more tablets are distributed and a stable internet connection is assured. So rabies serves as a prime example of One Health, emphasizing the need to collaborate transdisciplinarily, and the application will hopefully be tailored and piloted in Mali soon, once funding has come through again. And with promising results, we hope to, this application has promised to also be translated and used in other pertinent zoonoses as well as rabies. A big thanks to the Block Rabies team, to my supervisors, and the local team for welcoming me and teaching me. And we've been accepted to join the World One Health Congress, where we will be talking about the implementation work and our project in Cape Town in the 3rd week of September. So thank you very much for your attention. Please scan this QR code, and I'd be happy to talk with you after if you have any questions. Thank you.
Yes, we don't quite have time for questions. We're a little bit behind, but if you do have any questions, please feel free to chase up Jade during the lunch cocktail. And so, following on, second half of our graduate talks, please welcome to the stage Charusha Paul.
Good afternoon, everyone. Today I would like to talk about what I did during my master thesis during this IDO program. I worked on TR1, a stress granule protein, is a host factor for onion virus infection. So I would like to start with why this is important. When we hear about symptoms like joint pain, fever, headache, rashes, or nausea, we often think of diseases like dengue, malaria, or chikungunya causing them. But during an outbreak in 1959 in Uganda of Africa, a novel virus was isolated causing these overlapping symptoms. This virus was mostly spread by Anopheles mosquito and was named onion virus, upon its most prominent feature being joint pain, which was locally called onion. The epidemic of 1959 continued for 3 more years, spreading to different parts of Africa and causing over 2 million cases. Besides small outbreaks and sporadic cases, another epidemic in 1996 caused over 1 million more cases, which was limited to Uganda alone. Even though the endemic circulation of this virus is currently endemic to Africa, Recently, a report of an isolated event has been found in Germany due to traveling from the endemic region. This highlights the potential of global spread due to a vector virus competence development in this era of globalization. Further research on this virus and its mechanistic approach is necessary because we don't have a vaccine or therapeutic targeting it. Also, we lack knowledge of its potential as a virus to spread and cause rather severe diseases. Furthermore, we don't know what are the reservoirs which are holding this virus. Another interesting thing is that we think that these virus cases are rather an underestimation, not only because of its overlapping symptoms with different arboviral diseases, but also because it has an 85% sequence similarity with chikungunya virus. Chikungunya virus and Ongyong virus sequence similarity makes it difficult for identification by serology due to antibody cross-reactivity. Both chikungunya and Ongyong virus are parts of alpha viruses, which are enveloped viruses with a single-stranded RNA genome. The genomes are divided into two open reading frames, for the first one coding for the non-structural proteins, and the other one codes for the structural proteins. Working on the mechanistic approach of this virus and its molecular details could lead us to potential drug targets, which could be used to counter the virus, which are not only affecting Ongyong virus, but also its relatives as overall alpha viruses. Our group has been working a lot with novel factors of different alpha viruses, and during one such experiment, a human protein TIA1 came up as a host factor for chikungunya infection, as the knockdown of it could reduce chikungunya infection. But we still don't have any study which could show that the knockdown of this protein is related to Ongyong virus infection, which laid the foundation of my research. So let's see, what does this host protein do? This protein is an RNA-binding protein having 3 RNA recognition motifs and a Q-reach region which can bind to different proteins and interact. During normal condition, the cell has the distribution of this protein in the nucleus and the cytoplasm in an even manner. But when a cell is under stress condition, the cell tries to sequester all the mRNAs that are not needed for cell survival using these RNA-binding proteins like TIA1 and others to form these cytoplasmic granules called stress granules. These stress granules can then reduce the stress like viral infection by interferon signaling, regulation of viral and host gene expression, and also apoptotic regulation. But different viruses have found out ways to manipulate this stress granule regulation by induction of stress granule regulation, inhibition, or doing it in a temporary manner. But what is the dynamic of Ongyong virus in this stress granule regulation? Which don't know. Which brings us to the objective of my study, to investigate the involvement of TIA1 in stress granule regulation during Ongyong virus infection. I will briefly present my findings during the short period of time. I first started with establishment of the Ongyong virus infectious work in my lab. I had 2 infectious clones. One is the NSP3, which has a ZS green tag tagged to its non-structural protein 3, while the other one has a ZS green tag from the second subgenomic promoter. After effective in vitro transcription and electroporation, I was able to collect the virus as 24, 48, and 72 hours post-electroporation, getting high titers which peaked at 48 hours post-electroporation. Now that I have the virus, I wanted to answer if TIA1 was at all important for Ongyong virus infection as it was for chikungunya infection, for which I advanced with a knockdown approach in HEC293T TIM1 cells and a more relevant dermofibroblasts, because this is the cells which would be in the first target when a mosquito bites the patient. For this, I targeted the siRNAs on both of the cells, which would target the protein of interest like TIA1, and then infected the cells and followed by live cell analysis. This gave me the implications that when there was no knockdown of the cells within, no knockdown of the proteins within the cells, the normal levels of infection could be judged by the ZS green intensities represented in the left. While when TIA1 was knocked down, the intensities of this infection went far low, giving lower levels of infection. I could follow the ZS green over time, ZS green expression over time, and develop a growth curve, which is represented as a normal infection curve in gray, and the knockdown effects of TR1 in yellow. I took a closer look at 24 hours to find out that in both of the cell lines, HEC cells and the dermofibroblasts, the knockdown of TR1 was reducing the infection level greatly, while the knockdown of related stress granule proteins, which is TRL and G3BP1, was rather increasing the infection level, which gave me the conclusions that TR1 has proviral effects, while TRL and G3BP1 has antiviral effects on onion virus infection. I further validated my results using knockdown controls with qPCR at transcription knockdown and Western blot technique for proteomic knockdown levels. Now that I know that there is an effect, I wanted to judge if this effect was in replication, for which I used a luciferase-based transreplicon system. I had two replicons. The first one is the normal replicon only containing the non-structural proteins, making it replication efficient, while the other one has a mutation at the non-structural protein 4, which is the RNA-dependent RNA polymerase, making this replicon replication deficient. When either of these replicons were co-transfected into the HEC293TTM1 cells with a template plasmid, which beared the untranslated region of the onion virus 5', followed by luciferase genes, these NSP4 would recognize the untranslated region and transcribe the luciferase gene downstream, which could be read by the luciferase activity. Thus, by the luciferase activity would indirectly give us an indication of the replication ability of either of this replicon. So when I looked into the graphs, I found that the normal replicon abilities are demonstrated in red and blue, while the replication deficient abilities are represented in dotted lines. I compared my replication system with an already established replicon system in the VEV, which is demonstrated in the gray, and I saw that my replicons were not performing as good as VEV, but we knew that this was the systems which not always perform the best. I was happy with the fact that they gave stable reads at 24 and 48 hours and continued with my experiment, where I first knocked down the TR1 and other proteins in HEC293TTM1 cells, and then did this replicon assays and measured the luciferase to find that the replicon system would go down in expression when TR1 was knocked down, while TRL did not have an effect in replication, and G3BP1 had an increased effect upon knockdown. Thus, TR1 is a replication factor for onion virus, while G3BP1 blocks in replication. Now that I know that there is an effect in replication, I wanted to narrow it down if there is something to do with the stress granule regulation, for which I first took as a control chemical induction of stress granules with arsenite, where I saw that TR1 and G3BP proteins would co-localize, forming these granular patterns which are otherwise absent when the cells are in normal condition. I saw similar patterns of these granular structures when dermofibroblasts were infected with onion virus, and these granular structures would co-localize with the replication complex NSP3 and the double-stranded RNAs, but in different manner. We saw a complete co-localization of NSP3 and G3BPs, but only partial co-localization with its double-stranded RNA. I tried to do the stainings with the TR1 protein, but did not have time to look into it. But I saw that the G3BP granules that I had was rather consistent over time, and I could detect the granules as early as I could detect the proteins in the cells. So I did not have the TR1 stainings. I rather thought that maybe an in silico approach could give me an idea if TR1 was interacting with the replication complex, for which I tried to obtain and predict protein structures using AlphaFold, and then measured the quality assessment of these structures, and later I protein dock the TR1 with onion virus NSP3 and chikungunya NSP3 and interpreted their interaction. I could see that both of the NSP3s of chikungunya and onion virus has a scope of interaction with TR1, having similar contact sites and similar contact scopes, and mainly localizing to its hypervariable regions, but we still don't know the functions of this hypervariable region. But we could definitely predict that TR1 has an interaction scope with the replication complex. So I would like to summarize my findings. I first established the infectious work and the replicon system in the lab, which could be useful BSL-2 tools for working with different alpha viruses. I found out that the novel host factors, TR1 is proviral, while TRL and G3BP1 have antiviral effects. TR1 and G3BP1 effects were prominent in the replication, while TRL was not found to be contributing to the replication and indicates other possible outcomes. I found that G3BP granules are induced upon onion virus infection, and that TR1 has a scope of interaction with the non-structural protein 3. Now let's try to discuss why these findings are important, because we think that TR1 could help in stress granule formation, where it could accumulate all the necessary items for the replication of the virus, like the initiation factors, ribosomal subunits. But this hypothesis is opposed by the fact that two different proteins, G3BP1 and TRL, have an opposing effect in the same replication complex. This is not something which defines it. So we think that rather the TR1 would stabilize the viral RNA for replication, while the sequestration of the G3BPs are by the NSP3s, which would inhibit the stress granule formation. This is supported by the fact that the double-stranded RNAs would only partially co-localize with the G3BPs and allow the replication compartment to be separate. If this hypothesis is true, we could find out through further research, different antivirals to target the interaction of TR1 with replication complexes and double-stranded RNA. Also try to induce stress granules, as it would be a stress granule inhibiting protein. But to work on it, we still have a lot of things to do. First, confirming that if these granules that we see are stress granules, to look over the single kinetics of the single cells by live microscopy, confirming the effects by overexpression, and also looking into the interactome upon stress granule regulation. With this, I would like to thank everyone for your attention, particularly acknowledging my coordinators and peers from the masters, my team from THO Hanover, my supervisor and co-supervisor. Thank you very much.